when you step into an elevator and push the button, you expect it, you take it for granted really, that it'll take you to the right level. And when it doesn't, then you get concerned. You might be stuck somewhere, or you might be flying down the elevator shaft. Either way, it's a really big problem. There's another set of machinery that we take for granted, and that would be the protein machinery themselves. I study a family of proteins called G protein coupled receptors, and each has a specific function that's essential for our survival. One of these is metabotropic glutamate receptor 5, or mGlu5. mGlu5 controls the amount of glutamate in our brains, and this is really important for learning and memory. If you have too much glutamate in your brain, then you can get the hallucinations and illusions seen in schizophrenia. If you have too little glutamate, then what you'll have is the mental retardation we see in Fragile X syndrome. But thankfully, though, in that broken elevator, there is an override function, a key that allows you to control the elevator manually if it's not working correctly. But imagine you're in this broken elevator and you have hundreds of keys, and you don't know which one fits. You can try them one at a time, but this is going to take a lot of time that's very precious. If you only knew the size and shape of the key that you needed, this could save your life. Drugs that bind G protein coupled receptors work very similarly to these keys. If you could find a drug that fits perfectly within the binding pocket of a protein, it can override a protein that's not functioning correctly. If we could find a drug that binds mGlu5, this could really change the face of cognitive disorders. But the problem is, there are not just hundreds, but millions of chemical compounds. If we're going to bind a drug effectively, we need to know the size and the shape of that binding pocket. We can use X-ray technology to do this, but it's so difficult that we only know the structure of 2% of the 800 G protein coupled receptors in our body. mGlu5 is not one of them, so we end up testing one compound at a time. This is why it takes not only years, but $1.8 billion to generate just one drug compound. But what if we could use chemistry, physics, and our knowledge about protein structures and create a computational algorithm that could create for us a 3D model of our protein? We can then use the computer and that model and test which drugs can fit inside the model's binding pocket very quickly and easily. My lab has generated these algorithms. I've used them to model how a few drugs can bind mGlu5. One of them turns off mGlu5 and can be used to treat Fragile X syndrome. One turns it on and can be used to treat schizophrenia. But I'm not just stopping at mGlu5. My protocols can be used to generate the structures of all 800 GPCRs, G protein coupled receptors that we have, and change the face of disease. If you're facing a crisis, finding the right key can really save a life, and that's what my research is doing here. Thank you.